So hello, welcome back to the Science Shack. So we've got a new topic for today. This is an AS level topic, so part of the first year of A level study on genes to proteins. So specifically, how do genes get to be proteins? Now, I'm assuming for this that you'll remember the GCSE version of this that you did. If you don't remember that, then go back and watch the series of videos on how this works at the GCSE level, because we're going to add in a little bit more detail, of course, for A-level standard. Now, if you are a GCSE student watching this, well done, I salute your commitment. So, the first lesson in this is genes and the triplet code. So a little bit more information on how DNA actually gets read. So how DNA is deciphered, decoded by various proteins and by the ribosomes. Okay, and this is a bit of an overview style lesson. We're going to skim over a few things that we then loop back to later in the topic. So it's these bits of the syllabus we're looking to address. The gene is a base sequence of DNA. What does it do? That it can code for amino acids and some new stuff for A-level. And then obviously what the word locus means, and the idea of the genetic code being this triplet code that has rules and it has spellings and punctuation and grammar like any good language should. Okay, so here's our objectives. Firstly, describing the nature of the gene, and then explain how genes code for polypeptides in a sort of broad overview type of way, and that's what that picture down at the bottom left shows you. So before we start that though, an important rant, a rant I did at GCSE level with this, but it's an important thing to understand because it will stop you tripping up further down the road. Hopefully you got this at the beginning of your mitosis lessons, meiosis lessons, and in your genetics lessons. It's really important to get through your head because it's a mistake that A-level students and GCSE students make all the time. Kind of acceptable for GCSE students, not for A-level students. Okay, so your genetic material is packaged up nice and neatly. We're not going to talk about humans for now, into 46 little packets arranged into 23 pairs. Okay, those are pairs of chromosomes. Okay, each one of those chromosomes there comes from your mum and from your dad. Okay, now colours don't really matter, but one is the maternal chromosome and one is the paternal. And there are 23 of those pairs: pair 1, pair 2, pair 3, pair 4, pair 5, so on and so forth, right? But most of the time during a cell's life, you cannot see the chromosomes. The only time you can see the chromosomes is during replications, during mitosis and DNA replication. So what we've got here, look, these are also chromosomes. So this yellow object here is a chromosome. This cross-shaped yellow object here is a chromosome, right? Not a pair of chromosomes. This is a pair of chromosomes. This is a pair of chromosomes. This is one chromosome. Do not make that mistake. The only reason it looks like an X is because it has already gone through DNA replication. If you think about when in a cell's life this is, right? This is just before they go through mitosis, they split up. So DNA replication has already happened, right? The equivalent would be saying, I've got two cake recipes here. The implication being, I've got one way to make a chocolate cake and one way to make a vanilla sponge or something like that. That's two cake recipes, not one photocopied. That's I've got two copies of one cake recipe. That's the equivalent here. Okay? Please, please, please do not make that mistake. That's a pair of chromosomes. This is a pair of chromosomes. That is not a pair of chromosomes. That is one chromosome that's been through a bit of photocopying. Okay? Get it right. So, speaking of chromosomes, we're now going to zoom in on some chromosomes. We're going to have a look, what is the nature of a gene? So what do we mean by a gene? Okay, well, really, straight off the syllabus. Okay, the first point there, a gene is a base sequence of DNA that codes for the amino acid sequence of polypeptide. You already know that from GCSE, that genes, DNA codes for proteins. Okay, you know that. It's what we did at GCSE. But a little extra bit for A-level, the intermediate molecule that we kind of skimmed over at GCSE, the so-called RNA, can also be used to do stuff. Okay, so-called functional RNA. So DNA codes for mostly protein, mostly polypeptides, but also these functional RNAs. Okay, so that's one addition, one point. And we've got these ribosomal RNAs, transfer RNAs, which we're going to get to later in this topic and look a little bit at in this lesson. So how do genes control reactions in a cell? Or how do they actually control the cell? Again, covered at GCSE, a little recap. Well, they code for proteins. Proteins could be enzymes. Okay, enzymes are proteins, but not all proteins are enzymes. And enzymes control chemical reactions in cells through regulating metabolism. Okay, and then there's this word locus you have to know. So a locus is a place on a chromosome occupied by a specific gene. So if I went to that point on that chromosome in any individual person, I would find their version of the gene for making the same thing. Okay? And the way I always like to think of this is like a filing cabinet. So a chromosome is like a filing cabinet. The drawer and the position that the folder is back in that filing cabinet, that is basically the locus. Okay, and if I go to that place in two adjacent filing cabinets, I'll find instructions for making the same things. There might be different versions, right? They might tell me, one might tell me to make a chocolate cake, one might tell me to make a vanilla cake, as I said, but they are the same basic instructions, make a cake. Okay, and that same thing goes for eye colour, hair colour, and obviously as an A-level student, you will have had some introduction to inheritance, dominance and recessiveness from GCSE. That's where all that stuff comes in. So locus, important keyword, write it down, make sure you know what it means. The place on a chromosome occupied by a specific gene. Okay, so here's our overview. Now, we're going to get pretty detailed into this in our topic and throughout this topic. So I would recommend making sure you keep this picture close to hand or one like it. An overview of what's going on. What we're talking about is how information from the DNA here in the nucleus ends up via a messenger molecule and a ribosome becoming protein or polypeptide chain. Because we're going to get quite detailed about some of the minutiae here, and you will get lost if you don't keep perspective and context. Okay, so we'll come back to that picture a number of times. Right, so the first important molecule you do now need to know about, and I mentioned in a throwaway fashion at GCSE, but now it's required knowledge, is this messenger RNA molecule. Okay, what it does is it carries the message that codes for protein from the nucleus to the cytoplasm. Okay, they're single-stranded molecules, just long enough to contain one gene only. We're not going to go too much into the structure of RNA because it was covered in the nucleic acid topic, and we're going to come back round to it when we talk about comparisons between the different forms of nucleic acid. And this little picture, lovely picture here, is actually from Moderna. Them of vaccine fame, because of course a lot of vaccines for the coronavirus and various others. Super modern way of vaccinating people is to use this messenger molecule injected into a patient to signal their cells to produce an antigen that you're supposed to raise an immune response against. It's a very clever way of using mRNA. And the way they describe it is DNA is for the storage, stores instructions for proteins. mRNA is like the software. It's a temporary set of instructions for cells to make a protein. Okay, and then obviously that mRNA tells the ribosome how to put amino acids together to make proteins, which are for an application within the cell. Okay, thank you very much, Moderna, for that. They of course only have a short lifetime because cells need to change what they want to do, right? Because if they were making adrenaline, for example, they don't want to do that for a short amount of time, not for loads and loads of time. Okay, so mRNA is degraded after use, and if you know your differences between RNA and DNA from the last unit, you might see why that is. We're going to skim over that now and come back to it in another lesson. You also got another new molecule to learn, which is tRNA. Now at GCSE, you will have been told all about the idea that the ribosome just happens to know which amino acid goes with which triplet, right? Which triplet on this messenger molecule, an amino acid, and the ribosome somehow knows, yes, that's that one. Oh, that means proline. Brilliant, right? Well, actually, there's another molecule in there, which now you have to know at A level, called the tRNA, the transfer RNA, and it's literally an adapter. The little picture's there. Okay, so it's an adapter molecule. So it matches the amino acid to their codon. You can probably see where the amino acid and the codon are already. We call this a clover-leafed structure. It's a single-stranded RNA molecule that's folded over on itself. At one end, right, the base is ACC there. That's where the amino acid is attached. Okay, so it's like a little towing that amino acid in. And down here, you've got a thing called the anticodon, and it's that thing that binds onto the RNA, binds onto the messenger molecule. So if you're saying, how does the ribosome know which amino acid to pull in? Well, it's exactly the same as how DNA knows how to pair up. You know, A's with T's, or rather U's, and C's with G's. Okay, so those things pair up very nicely. That is like the adapter molecule between the two. Okay, so we'll come back to tRNAs in more detail in another lesson when we look at them in comparison to mRNA, rRNA and DNA, but for now, just now you know new information, tRNA exists. So, if we're going to look at an overview of how protein synthesis works, you start, of course, with the code for the protein on the basis of the DNA. You then get a process called transcription, which is copying out. Now, depending on who your GCSE teacher was, they might have mentioned the words transcription to you. It's transcribed into this sort of messenger molecule, which we now know is called mRNA. Right? At this point, though, however, it still needs a bit of work done to it, so it's called pre-mRNA. That pre-mRNA is spliced, so bits of it are cut out. Don't worry for now about why. We do a whole lesson on splicing and processing in the transcription process to make what's called mature mRNA, and that is then translated. Right? mRNA is used as a template on the ribosome to add amino acids in the correct order, using tRNAs to help, as previously described. Okay, and then we get protein. Happy days. Okay, so that's our overview of what we're trying to go through here. So let's define a few more terms in this lesson. Triplets and codons, what we mean by the two. Difference is subtle, but it's there. A triplet, we say, is 3 bases, usually the term we use for 3 bases on DNA, and a codon is those 3 complementary bases on RNA. So as you can see here, you've got the RNA there, look, the sequence coming down. So GCU is 1 codon. ACG is the second codon. GAG is the third codon, and so on and so forth. So a triplet on DNA, which is what you would have learnt at GCSE, the triplet code, right, codes initially for a codon on the mRNA, and that tells the ribosome which amino acid to put in. Okay, so this group of 3 bases on the mRNA coding for amino acid we call a codon. There are 20 amino acids and only 4 different bases. So hopefully you can see why we need a code of 3 lots, because we couldn't use 2 or 1, there wouldn't be enough combinations to get up to 20. Okay, so bases are read in combination in groups of 3. There are 64 possible combinations. Obviously that is more combinations than there are amino acids, and we'll get to what happens in the case of repeats in a little while. Okay, and this bit here, right, the meaning of each of these 64 codons is something that has only recently been discovered by humanity, and it is a genuine privilege to be able to share this information with you, because it's one of the most powerful pieces of modern biology that we can actually share. It is the genetic code. Okay, there it is. It's in all the textbooks, and that's all the meanings of all of the different codons. So that's the triplets on RNA, not the DNA, and you will know that because there's U's in there, not T's. Okay, so it's the meaning of all the different codons and what they produce in terms of an amino acid. Using that, you can not only decode any protein you like and work backwards to what its RNA or its DNA should look like. You could also go forwards from any bit of DNA and work out what protein it should form. And you can even design your own piece of DNA to produce a custom protein. Okay, now that is obviously an incredibly complicated thing to do, but that is the world in which we now live, and it's why it's such an exciting time to be a biologist. Okay, so you often see sometimes written like that. I mean, I prefer the square version. In some textbooks you see it written like that, where you've got your first base in the middle. So if you want to look at what CCC was, you'd go C here, out 1, second base is C, out 1, third base is C. That's proline. Okay, I personally prefer the square here, where we have the first base down the side, the second base along the top, and the third base down the side. So for example, CGU is arginine. Okay, right, now that's basically spelling. What I've given you there in terms of the genetic code is the dictionary, all of the words that can be used to make proteins, all of the different words that can use to spell out sentences that are proteins. The genetic code, however, does have rules as well. It has punctuation, it has grammar, it has ways of working. So it is, for first rule, and these are worth jotting down, it is a linear code. It's only read in one direction, and I've got a handy book just over here for this purpose. Okay, now if I was reading this book, what's the first thing I would have to know? Well, I would have to know how a book works. Right? If I was an alien who was dropped down from space, okay, very appropriately, The Voyage of Charles Darwin, I would have to know that in English, I open a book like that, and I start at the top left, and I read along to the right, and I work my way down. Okay, not the same in Arabic, not the same in classic Chinese, where they go different directions. DNA has a similar rule. you read from the 5' end to the 3' end along the mRNA molecule. Okay, so second one, the code is degenerate. And what we mean by degenerate is that more than one codon codes for the same amino acid. Right? So CCA, CCC, CCG, and CCU all code for prolines. How we get around that idea that there are 64 combinations, okay, 64 different combinations, and there are only 20 amino acids. I always liken this in English, it's like synonyms. Okay, lots of different spellings mean the same thing, don't they? I mean, there's lots of synonyms. You can get thesaurus out. Many of you do so when you're trying to write your personal statements to a very poor effect. Or it's very much like the current fashion for girls' names all ending in E, right? So I use Ellie as an example, as quite a good example, because as you might notice here, look, CCC, CCA, CCC, CCG, we've changed the last base, but not the first two. So the first ones are more important than the third in specifying what the amino acid is. Okay, so what's happening there is it's like Ellie could be spelt E-double-L-Y, E-double-L-I-E, E-double-L-I, E-double-L-E-E if you're being particularly awkward, or the completely inexcusable E-double-L line with a little heart over the top. No excuse for that, right? So, but if you change the first letter to Melly, belly, then of course it changes the sound of the word. Okay, very similar. Last based, less important than the first. So it's linear, it's degenerate. The first two bases are more important than the last, and it's non-overlapping. So, of course, in the English language, we have a thing called space bar, right? We have a space to signify this is the end of one word and the beginning of another. We don't have that in DNA. What we have is that every single word, or codon, is exactly the same length. So every word is 3 letters long, so you never get one base used in more than one codon, okay? It's 3, then 3, then 3, then 3. So the genetic code is a linear, degenerate, non-overlapping code. Okay? So that's our basic rules of how you read it. Every language needs punctuation, and there are a couple of punctuation marks you need to know about at this stage. The first one is the start signal. Okay? That's like a capital letter. Every protein starts pretty much with AUG. That's how the ribosome knows which bit of the RNA to bind onto and to start doing translation. Okay, so AUG, it codes for the amino acid methionine, is like our capital letter. The ribosome will run along, find an AUG, and start its translation from there. Okay, so there have also got to be full stops. So you might have noticed the eagle-eyed amongst you when looking at that genetic code, there were 3 codons, 3 triplets that say stop, right? Those are UUA, UAG, and UGA. They do not code in nature for an amino acid, and it has been that way for 4-point-something billion years. Okay, as the best estimates of when life emerged, just over 4 billion years ago at the moment. And these stop signals have been absolutely sacrosanct. There's been no amino acid, no tRNA that can drag an amino acid onto that codon until your lifetime. A few years ago, a group of scientists managed to work out they could design a tRNA to be complementary to one of these and drag in a completely artificial amino acid. Now that is hacking the machine code of life that's existed unchanged for about 4 billion years. And you're living through that moment in history, which is why it is an incredible privilege to be able to actually share this knowledge with you. Okay? And perhaps the most profound thing, the most profound rule of the genetic code is that it is entirely universal. Okay? Apart from organisms like RNA viruses that don't use DNA at all, right? And they have weird exceptions. The code is universal. The same base sequence always codes for the same amino acid regardless of the species. Regardless. Doesn't matter. Okay? And that is mind-blowing for 2 reasons. First is that means we can actually now cut bits of DNA out of one organism, take them from one organism with relative ease and splice them into another organism and know that that organism will understand that DNA and be able to use it. which is an incredible fact. That's basically the basis of genetic engineering. It's why we've got goats that can produce spider silk wandering around. It's why we can knock up glow-in-the-dark bacteria in an afternoon in the lab with a bunch of Year 12 students. Okay, it's not difficult. Okay, it's why insulin is now manufactured using bacteria rather than blended up pig pancreases, and so on and so forth. Okay, so that is one thing that is incredibly important, incredibly profound. It's going to have massive impacts on how we edited our own code and our own genes. And even more profoundly than that, and I think this is the most wondrous thing about this fact, is it is irrefutable proof that we are all related. The fact that the basic machinery of life works the same, whether you're a bacteria, whether you're a plant, an animal, a fungi, whatever, doesn't matter, right, is absolute proof that we are all related. We all share a common ancestor, a common origin. And of course, the more genetically different we are, the further back that common ancestor is. Okay, and that's led to incredible discoveries in the fields of phylogenetics and understanding our relatedness and classifying life. So that amazingly constructed genetic tree of life, I'm going to leave you with there, is one of the most profound implications of our knowledge of DNA. Okay, and it's why I cannot wait to do the rest of this topic. Okay, so I'll see you next lesson. I hope that was all right. Bye bye.